Hi everyone, happy Tuesday. Welcome to today's very special presentation of MDF's Meet the DM Drug Developer. We are talking with Avidity Biosciences today. They are going to be discussing AOC 1001 MARINA top-line data from the Phase 1/2 clinical trial for DM1. Just a couple of things about the foundation. We envision a world with treatments and a cure for myotonic dystrophy. Our mission is community care and a cure. We support and connect the myotonic dystrophy community. We provide resources and advocate for care, and we accelerate research towards treatments and a cure. A reminder of some of the resources and support available from MDF. Our toolkits and publications, including clinical care recommendations and guidelines, are found at myotonic.org/toolkits-publications. Support program information can be found at myotonic.org/find-support. Our calendar of events is at myotonic.org/calendar/month. And our digital academy, which includes all of our past presentations, including the Meet the DM Drug Developer series, can be found on the digital academy at myotonic.org/digital-academy. Today's presentation is being recorded and will be added to the Digital Academy as soon as it is available. A reminder about the upcoming Meet the DM Drug Developer sessions. June 9th, we have Artex Biotech. July 7th, AMO Pharma. Avidity will be back to join us on the 4th of August. And then we hope to see everyone in Washington, D.C., September 7th through 9th for our annual conference. You can find more information about our conference at myotonic.org/conference. We will be holding the first of a 3-day conference, which is focused on advocacy at Capitol Hill for that first day. So we are very excited and hope you will join us to talk with representatives and senators. Okay, today's Avidity Biosciences team is Sarah Boyce, the Avidity President and CEO, Steve Hughes, Avidity's Chief Medical Officer, and Nick Johnson, who is the lead principal investigator for Avidity's MARINA trial. As a reminder, many of you have submitted questions in advance. Thank you so much. The Avidity team has done their best to integrate the responses into their presentation. If you have additional questions or follow-up questions, you can ask those live. Just open the questions tab, type your question and click send, and it will go to this team. If you're on a smartphone, you can click on the question mark icon at the top of the screen, type your question and click send. If today's program speaks to you, please consider showing your appreciation with a donation to MDF. You can find the donation page at myotonic.org/donate. Okay, it is my great pleasure to welcome the Avidity Biosciences team. Thank you so much for joining us today.
Thank you so much, Tanya, and thank you for everyone for joining us today in what is incredibly exciting news to share with everyone on the call, which is the results of our top-line data from our Phase 1/2 study in myotonic dystrophy. I can't see my slides at the moment, so I'm hoping that somehow the slides will magically appear. But just before I begin, from an aspect of, we're using terminology that I know not everybody may be familiar with. So firstly, what does top-line data mean? So when you complete a Phase 1/2 study, which is the first 2 phases in the drug development process, you get a lot of data that comes in as you close out the study. When we use the term top line, that's because it's the first set of data that's coming in-house that we're able to analyze, make sure sort of we do all our quality controls and then present. But it's also called top line because there is still more data to come. And typically, all of that additional data will ultimately form into what is a publication, which has a full review of all of the data and findings from the study. So what you're seeing today is that initial data set, but also from an aspect of the most exciting and the most immediate data set in terms of looking at the impact that AOC 1001 is making in the clinic. During the, if we can go to the next slide, please. During the course of this presentation, I will be making what's called forward-looking statements. This is part of being a publicly traded company, and you'll see sort of in these slides, this is in detail of our forward-looking statements, and I invite people to read it at your leisure should you wish to do so. So in terms of who is Avidity and what we're trying to do, we are deeply committed as a team to improving the lives of people impacted by diseases. And this in particular is including myotonic dystrophy through the development of new drugs. AOC 1001 is the first of our drugs to enter into the clinic, and the first of our drugs where we're seeing top-line data coming out from a phase 1/2 study. At its simplest form, we are a group of people trying to bring great science and ultimately drugs to other people. That is at its simplest form. From an aspect of any significant thing in life, it always takes a village, and we are part of a community and a part of the myotonic dystrophy community. And with that, we are extremely grateful to the contribution that has paved the way to where we are today. And if we could go to the next slide, please. That contribution and what has paved the way to the MARINA trial is very much the work of the Myotonic Dystrophy Foundation, as well as the Myotonic Dystrophy Clinical Research Network, in having a network already established for a rare disease for when a program comes into the clinic. Of particular importance has been that work of the foundation in funding the NDM1 study and establishing biomarkers and clinical endpoints for myotonic dystrophy type 1. This is incredibly important from an aspect as then as a company we're looking to design clinical programs. And this has been the work of many patient advocacy organizations, and including the Myotonic Dystrophy Foundation. The other aspect is one of them, sort of the core aspect of this is the community. The myotonic dystrophy community for people who are living with myotonic dystrophy, as well as their families. We know that a rare disease doesn't just impact the individual living with the disease, but the whole family, as well as also the caregivers who are involved in supporting those people who are living with myotonic dystrophy. We are incredibly grateful to this community for bringing us where we are today, and look forward to our long-term partnership as we look to move further forward in the clinical development process. One of the other aspects is participating in a clinical trial, particularly a phase 1/2 clinical trial with the new technology. It's an incredible gift that those participants in the study have given to the community. This simply would not have been possible without each of the participants who enrolled in the study taking part, and their families in supporting that, supporting the visits to the sites. This is also a study where participants in the study had muscle biopsies. Having a muscle biopsy is not the most pleasant process in the world by any means, and for people to sign up and volunteer to do that, to move science forward for their community. And what you'll see how that has enabled and really deepened our understanding of myotonic dystrophy, as well as also the potential for a drug like AOC 1001. There's also an aspect that it is an individual who participates in a clinical trial. But it is a family in the village around each of our participants who is also so involved. And I also want to recognize the contribution of families as part of that. Also recognizing the investigators that we work with, who really are incredible. And it's investigators like Nick Johnson, but also all of the team at those sites as well. This has taken time, this has taken commitment, and also longer term continued contributions and partnership, both in the MARINA study. So the MARINA study is what we're talking about today. That is the phase 1/2 study. And then also what is known as the MARINA OLE study. So an OLE is an open label extension study. So when a participant completed the MARINA study, which is a 6-month study, they then had the option to enroll into the open label extension study where they could continue to receive drug. And for the participants who were randomized to have placebo in the MARINA study, they were able to receive drug in the MARINA open label extension study. So in terms of moving to the top line data, I'm going to cover kind of like the highlights of what was observed. And if we can move to the next slide, please. This was really incredibly exciting for us here at Avidity, and I think for the investigators that we've been working with, and hopefully also for you as a community. What we were able to show from Topline, so that's the first readout from a phase 1/2 study, which is the first studies that you typically do in drug development. The next stage is a phase 3 or something that is called a pivotal study, then leading to registration. But in this phase 1/2 MARINA study, we saw disease modification via functional improvement within the doses that we had identified for this study. And in terms of from an overall perspective, AOC 1001 demonstrated directional improvements in myotonia, in strength, as well as early signs of mobility. So these are things like the 10-meter walk run time in a 6-month period. So actually in a pretty short period of time to be able to show these directional improvements. These improvements were followed by the knockdown of the DMPK gene, and then also the splicing changes that then occurred as a result of that. And this was all very consistent with our understanding of the mechanism of disease. And again, that understanding of the mechanism of disease is part of this community's work in building that. One of the things when you do a phase 1/2 study is you're looking to identify what your dose is in terms of being able to take forward into your next study. And we were able to do that, identifying a dose range in the 2 to 4 milligram per kilogram range. And one of the other things that we observed in this study, and you'll see this in the data, is that for the 2 milligram per kilogram, so people who are enrolled into that arm of the study, we need to dose more frequently. So we were dosing every 12 weeks, and we need to increase that frequency. One of the other aspects was also having a favorable safety and tolerability profile to move forward. So one of the aspects is what's next. And a key area at this stage in drug development is around aligning with the health authorities on a path forward. And that's what the team here at Avidity is working very hard in relation to doing. With regards to our goals for the day, and if we can go to the next slide, please. So it's sharing the results of the MARINA study, both in terms of safety, biomarkers. So the biomarkers are those signs of biological activity that will then, you hope translate into functional data. And the biomarkers that we'll be looking at is knockdown of the gene, as well as also impact on splicing changes. So the downstream elements of knocking down the gene, as well as also looking at functional data. So functional data, you'll see a look at a range of measures. And one of the aspects that has us very excited is the consistency in those early movements in those range of functional measures. As part of our style, in general, as a company, we love talking about our science and our programs. And also, we're very conscious that you may have many questions. So we're looking to be able to answer your questions in the Q&A session. With regards to today's agenda, I'm now going to hand over to Steve Hughes, who's going to take you through both what AOC 1001 is, the MARINA clinical trial, and he will then hand to Dr. Nicholas Johnson, who is the lead investigator for the MARINA study, and he'll share with you the top-line data from the MARINA trial. And then from there, we'll go to I'll then make some closing remarks, and then we'll go to a Q&A session. So without further ado, I would like to hand over to my colleague, Dr. Steve Hughes, who is the Chief Medical Officer at Avidity. Steve.
There I am. Thank you, Sarah. Hello, everyone, and thanks for your attention. As Sarah said, I'm going to tell you a little bit about our antibody oligonucleotide conjugate that we're using to treat myotonic dystrophy type 1, which is AOC 1001. And give an introduction into to tee up Dr. Johnson's presentation, where he's going to walk you through our very exciting top-line data. So if we can move on to the next slide, and I apologise, I'll be needing to have some water. I've got a bit of a frog in my throat today. So this is a schematic of AOC 1001. And we'll come back to this in just a few moments after talking a bit about the disease, because the various parts of the drug each have different functions. So this is an antibody oligonucleotide conjugate. The turquoisey color, big thing to the left-hand side there is the antibody. And we'll talk about that in a subsequent slide. And then the red-coloured piece on the right-hand side is the oligonucleotide. And it's actually the oligonucleotide that is the active part of this drug that is targeting the underlying cause of the disease, whereas the antibody is used to direct the oligonucleotide component to the muscle cells. So if we can move to the next slide. So before getting a little bit more into the drug, it's helpful, I think, to just do a recap on the underlying cause of the disease, because from the underlying cause of the disease, an understanding of that, it would makes for a better understanding of how the drug works. So DM1 is caused by a mutation in the DMPK gene. So that's the myotonic dystrophy protein kinase gene. And if you remember, our genetic code, which is essentially the blueprint for how every single cell in our body works, is held in DNA. And within the DNA in our cells, individual proteins are encoded by genes. And genes are strands of DNA that basically give the blueprint for each individual protein that we need for our bodies to work. And the genes that are held in the DNA genetic code, they don't directly make the proteins. There's an intermediate, and that intermediate is RNA, and specifically messenger RNA. So what happens is the blueprint, which is the DNA, reads off into RNA, and that RNA then provides the template to make the proteins. And in myotonic dystrophy type 1, the RNA that's made has a very long repeat tract in it, and a repeat of 3 different nucleotides. So the nucleotides are the individual coding elements that tell the cell what protein to make. And within the mutant DMPK messenger RNA, there's a very long tract of CUG, where you get CUG, 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 on and on and on. Normally, in normal people's cells, we know we may have 30 or so of these repeats, but in myotonic dystrophy type 1, these repeats can be hundreds or even thousands of CUGs in length. And what those CUGs do is they bind up what's called splicing proteins or splicing factors. And those splicing factors are needed to make normal messenger RNA for all of the other proteins that the body uses. So by binding up or sequestering the splicing factors, the individual cells within the body are unable to make a whole host of different normal proteins. And because it affects every single cell within the body, you can imagine that this is what might be called a multisystemic disease, or a disease that affects multiple organ systems within the body. And you can see here a whole range of the different body systems that can be affected by myotonic dystrophy type 1. So it really is a disease that can have multiple different manifestations within individuals. If we click to the next slide, many of these manifestations, however, are from muscle. So just before we move into the muscle part, I think it's important to remind everyone that our drug AOC 1001 doesn't get into the brain. So the cognitive elements of myotonic dystrophy, we're not anticipating being able to correct with AOC 1001. But many of the other symptoms of the disease are actually muscular. So the respiratory system manifestations, the skeletal muscle, these are the muscles that you use to walk around and go about your daily business. Cardiovascular system, this is the heart and the conducting system within the heart. And then finally, the gastrointestinal system. All of these produce multiple symptoms and difficulties during the course of this disease. And our monoclonal antibody component of the antibody oligonucleotide conjugate delivers the siRNA or the oligonucleotide component into all of these different muscle systems, and this is where we're expecting to see significant impacts on the disease over time. So if we can move to the next slide. So back to the individual components of the drug. As you'll recall in, I think, my first slide, I said the drug consists of an antibody and it consists of an oligonucleotide, or here we say oligo for short. And those two pieces are bound together to make an antibody oligonucleotide conjugate. Antibodies aren't the only way of targeting oligonucleotides into muscle cells. Other companies are using different modalities, but we made a very deliberate choice when we came to design AOC 1001 in choosing an antibody. And there are a number of reasons for that. Antibodies are naturally occurring proteins that are made from by the body to fight off infection. So the body's used to seeing them. And also many antibodies have been used for other therapeutic modalities, both in cancer and in immune diseases and various other diseases. And there are now over 100 antibody type class of drugs that have been licensed by FDA. So the safety profile of antibodies is very well understood. And also, because so many antibodies have been brought forward into the clinic, and antibodies as a therapeutic modality have been around for pretty much 3 decades now. So a lot is known about them, and also a lot is known about how to manufacture them for therapeutic applications. And then the oligonucleotide component of our AOC 1001 is actually a small interfering RNA, or siRNA. And siRNAs are also well understood. They've been studied in the clinic for many years in clinical trials, and there are also now several small interfering RNA drugs that have been licensed by FDA for various different rare diseases. So how they behave within the body is very well understood. How we manufacture them is very well understood, and they have a good track record as a class in terms of safety. If we can move to the next slide, please. So now down to the mechanism. So oligonucleotides all are not created equal. And oligonucleotide just means that it's a small strand of individual nucleotides that have been joined together. So the oligo means a small length of, and then the nucleotide means individual nucleotides joined together. Many different types of oligonucleotide modalities that can be used for therapeutic purposes. For AOC 1001, we use what's called siRNA, or small interfering RNA. Similar to antibodies, this is a naturally occurring mechanism within the body used to fight infections within the cell, and it's a very specific way of targeting messenger RNA within the cell. So we can engineer the small interfering RNA to very specifically target a certain messenger RNA, and it works by basically cleaving or cutting the messenger RNA, so the messenger RNA then is degraded, and in the case of a toxic RNA can't cause any problems, and in the case of where a messenger RNA is making a toxic protein, you prevent the protein from being produced. So in this instance, we're looking to cleave the DMPK messenger RNA, so the toxic RNA that is the underlying cause of this disease, and in chopping that toxic messenger RNA up, we prevent it from being toxic, and therefore hopefully prevent all of the downstream consequences of that RNA on the abnormal splicing and the abnormal proteins that occur as a consequence. So then wrapping it all up, we see that we give the antibody oligonucleotide conjugate, and it's given as an intravenous infusion. That distributes throughout the body. It doesn't just get into muscle, it gets into many other different tissue types, just not into the brain. It gets taken up into individual cells, and in the muscle cells it will find its abnormal RNA, and it will cleave that RNA and prevent that RNA from causing problems in the disease. Okay, so if we can move to the next slide. Right, so now after the heavy stuff, we're going to get into the MARINA and the MARINA open label extension clinical trial, and I'm just going to give you a brief overview of what those trials look like in terms of what happens to the individual participants within the study, and then at the end of that I'll be handing over to Dr. Johnson. So if we can move to the next slide. So before I get into the study itself, just to remind everybody that AOC 1001 is currently on a partial clinical hold by FDA that came into effect towards the end of last year in response to a significant adverse event that was experienced, unfortunately, by one of the participants at the 4 milligram per kilogram dose in the MARINA study. There's a lot of technical medical language in what we have on this slide, but essentially the participant had a very rare type of stroke within an area of the brain called the thalamus, and that resulted in the participant having visual impairment and also some memory impairment as well, and unfortunately the visual symptoms of this participant have not improved and are likely to be permanent at this point, whereas the memory symptoms have improved. We conducted a very extensive investigation as the sponsor for the clinical trial, in collaboration with the investigators in the clinical trial, and also in collaboration with the patient and the patient's family. And that involved the patient having to have a number of additional scans and other tests to try to get to the bottom of what the cause for the event could be. And this meant a lot of going backwards and forwards to clinic to have various tests, and really we're very grateful to the patient and to the patient's family that they were able to help us get through this investigation of what was a very difficult time. the bottom line was, as we were not able to find any plausible linkage to the drug or to the transfer in receptor-mediated mechanism of uptake of the drug into the cells, or to the mechanism of action in terms of knocking down mutant DMPK. And so we're left with a bit of a conundrum in that we can't explain the how the event occurred. We've only been able to explain what the event was, that it was a very rare type of stroke. We're still in an ongoing dialogue with FDA about this event and about how best to move forward, and we'll come back and provide more information as soon as we reach a conclusion of that process. Turning back to the MARINA study now. So the initial study, which was MARINA, that's concluded. We wrapped that up a few weeks ago now, and all of the participants, except for one, have rolled over, or actually except for two. The participant that experienced the serious adverse event that resulted in the partial clinical hold isn't receiving further doses of the drug, and so has not rolled over into the open label extension study. And there's one other participant that is currently between studies. They finished the MARINA, and we're hoping that they'll roll over into the open label extension pretty soon. And as many of you will know, we presented the top-line data from the MARINA study, and this is the data that Dr. Johnson's going to share with you very soon at the American Academy of Neurology annual meeting just a few days ago. So just to orient you, and Sarah hinted at this a little bit earlier, top-line data, what does top-line data mean? So within a clinical trial, typically you're collecting many hundreds, if not thousands, of different data points. And when the study winds up and you're looking to look at the data, there isn't enough time for the biostatisticians to pull everything together to get all of the data out of the study in one go, unless you want to wait many, many weeks to get the data. So what we tend to do is to prioritize certain of the outputs that we want to see early, and they get busy with the computer programming that's necessary to look at those outputs, and so we see those first. So those would typically include safety and some of your key efficacy endpoints. And then while we're looking at and perusing that data, they're busily programming the other outputs. So the data that Dr. Johnson's going to present today, and the data that we presented at the AAN meeting, isn't all of the data from the clinical trial. It's the data that we deemed to be the most important for us to see first, and the data that we felt that was most important to share with the DM1 community and with the DM1 treaters first. So finally on this slide, the MARINA open label extension study remains ongoing, and we're looking forward to being able to share the first data cut from that study towards the end of this year. So could we move to the next slide, please? So this is a schematic of the MARINA study, which is on the left, and the MARINA open label extension study, which is on the right. It's a little bit busy, but it's really here to orient you to the results that Nick Johnson will be presenting in just a second. So if we focus on the left-hand side of the slide, this is the MARINA study. So that's the study that's now concluded. And this study was divided into basically two components, a single dose cohort and multiple dose cohorts. So the single dose cohort is the A cohort, and the multiple dose cohorts are the B cohort. Single dose was 1 milligram per kilogram, and the reason why that was a single dose is because we felt, as we went into the study, based upon our data in animals, that 1 milligram per kilogram was kind of at the start of the therapeutic range, and so we didn't want to continue exposing participants to what may be a marginally therapeutic dose for an extended period of time. So those patients had a single dose, and then they were followed up, and then subsequently were allowed to enroll into the open label extension at 2 milligrams per kilogram. And then in the B cohort, each participant received 3 doses. They received their first dose on day 0, and then they had what we call a booster dose at 6 weeks, and then another dose 6 weeks after that. And then they were followed up for 3 months in the MARINA study, and rolled over into the open label extension study, where they would then be dosed every 3 months. Although after the first dose, they received, the placebo patients received a booster, so that they were treated in the same way as the participants that received active drug in the main study. Participants were randomized 3 to 1, with, either to active drug or placebo. So that meant for every 4 patients that went into the study, 3 received active drug and 1 received placebo. And then as the placebos roll into the open label extension, that's where they start to receive active drug. So out of these cohorts, the participants in cohort A and cohort B1 go into the open label extension at 2 milligrams per kilogram, and participants that are in cohort B2, which is the 4 milligram per kilogram cohort, they roll into the open label extension at 4 milligrams per kilogram, although the placebos in cohort B2 go in at 2 milligrams per kilogram, and that was because of the restrictions of the partial clinical hold. The blue triangles that you see on each of these show the timing of the biopsies. So all participants within this study and within the MARINA open label extension study are having biopsies taken of their tibialis anterior muscle, which is the muscle on the front of the shin. That's responsible for moving your foot upwards. So for the A cohort, they received biopsy prior to receiving drug, and then at 6 weeks and 13 weeks. And then in the multiple dose cohorts, they have a biopsy at baseline at 13 weeks and then at 6 months. So the only other thing really to point out on this slide is the middle column, which shows the number of participants that were on placebo or active drug in the MARINA study. And then, as I mentioned earlier, all of those participants then receive active drug in the open label extension. Can we roll? So this is a slide that we used for our what we call the preliminary analysis. So within clinical trials, certainly early stage clinical trials, it's very usual to not just wait till the end of the study before looking at the data. And to have a look at the data along the way. And we built this analysis in primarily really because we were doing a lot of novel work on the muscle biopsies, and to make sure that the assays that we were using were performing as we expected, and to give us really a good idea that everything was on track for the final analysis of the study. But in going through the motions of pressure testing all of the individual pieces that would lead into the final analysis, it did make for a very interesting opportunity to actually look at the data and interpret it, and then to be able to share it with the outside world. And we did this back in December when we shared our preliminary analysis. And this was really focused on the biomarkers primarily, and on the whether there was or wasn't drug in the muscle. Although we did show some videos of some participants in the 1 and the 2 milligram per kilogram cohorts. We showed their what we call the video hand opening time, which is a measure of their myotonia, and we'll share those videos with you again in just a minute in Dr. Johnson's section. So at this preliminary analysis time point, we were looking at the muscle biopsies primarily. And we looked just at the 1 milligram per kilogram and the 2 milligram per kilogram cohorts. We hadn't completed the 4 milligram per kilogram cohort at that point in time. And what we were able to show was for the first time, an RNA therapeutic had been successfully targeted to the muscle. So we were able to measure the cargo, this small interfering RNA component of our antibody oligonucleotide conjugate. We could look at how much of that was in the muscle, and we were able to show dose-dependent increases. 100% of the participants that were treated with AOC 1001 showed reduction in the DMPK messenger RNA, and overall across the 2 cohorts there was a mean reduction of about 45%. And we were also able to show improvements in splicing in a panel of 22 different genes that we were using to assess correction of splicing. When we narrowed that down to just a 4-gene panel that looks at muscle-specific genes, that improvement increased to 31%. And so really this showed us proof of mechanism for the platform, in that we showed we were able to get drug into the muscle cells. We showed that when the drug was in the muscle cell, it engaged with its target. In other words, it reduced the DMPK messenger RNA. And we also showed that reducing the DMPK messenger RNA had the anticipated effect of improving splicing. And it's the splicing that is the underlying cause of all of the different clinical manifestations of the disease. Now, since then, we've done the top-line analysis, and this top-line analysis includes the 4 milligram per kilogram data from the MARINA study. And a part of that analysis, we have confirmed again proof of mechanism for the platform. So once again, we've confirmed that we're able to get the drug into the muscle tissues. We've confirmed that we're able to knock down the MPK messenger RNA, and that we see knockdown in all of the participants that are treated with the active drug. And we've also seen that the reduction in the DMPK messenger RNA results in the anticipated improvements in the splicing. This time, in addition to the 22 gene panel, we also looked at a panel of over 1,000 different genes, and showed that we were able to correct splicing, not just in the 22 gene panel, but across hundreds, in fact, over 1,000 different genes. And at this analysis, we've not only looked at a couple of the videos for the hand opening time, we've looked at and assessed in a quantitative way the hand opening time for all of the patients in all of the cohorts. And also we've looked at a number of other functional measures as well. And we've seen very consistent responses across all of the functional measures in terms of improvement, which is great. And this, for a first in human study, this is really quite remarkable to see improvements in everything. Normally, you might see improvements in maybe one or two things. We actually weren't expecting to see improvement in functional measures at all in a 6-month time point, because with neuromuscular diseases, the conventional wisdom is that these sort of endpoints take a year or more to change. And so we were absolutely delighted when we could see improvements. And in fact, we start to see separation as early as 3 months after the drug is started. So really after only a couple of doses of AOC 1001, we're starting to see improvements. So if we can move to the next slide. Okay, so I think this is almost my last slide now. So Dr. Johnson's going to be presenting results of a number of different clinical measures of the disease. And so what I wanted to do was to just walk you through how each of these individual tests is performed, so that you can appreciate the data more when you see it. So if we start in the top left with the video hand opening time. So this is to show changes in myotonia. So as you all know, myotonia is a difficulty with relaxing muscles, and is one of the cardinal symptoms of myotonic dystrophy. It's in the title. Myotonic comes from the myotonia symptom of the disease. And what we ask the participants to do here is to make a fist, and you'll see this in the videos in just a moment, and squeeze as tightly as they can, and then try to open their hand as quickly as possible. We video this. And then the videos go to independent, we call them adjudicators. So we have 2 people score the length of time it takes to open the hand in seconds. And what we're looking at is the time for the middle finger to go down to the flat surface. And we can see change from baseline. So in other words, what is the improvement from the first recording that you have before you take the drug, to subsequent recordings that you have after you've taken the drug. And this gives us a very clear measure of myotonia, but also it gives us an idea of how hand function might be improved. And dexterity and hand function is obviously very important for going about daily activities. And so this is a good indicator of what we might see in terms of improvement in patients' ability to go about their activities of daily living, we call them. Then on the top right, we're also looking at muscle strength. And here what we do is we don't just look at the muscle strength improvements of any single muscle, although we do look at single muscles, but we aggregate them into what we call a composite score. So we actually look at 6 different muscle groups, 3 in the lower limb, 3 in the upper limb, and we measure the improvement in muscle strength. So what the participants are asked to do is essentially contract the muscle group against a pressure transducer. And the pressure transducer measures the maximum force that the muscle is able to exert when it's contracting, and we express that as a % of normal. So the normative values by age and by sex for each of these different muscle strength measures, and so that allows us to express for each participant their muscle strength as a % of what you would expect, and then we aggregate all of that into an overall score as a percentage of normal. So the 6 tests that we do are hand grip, elbow extension and flexion, knee extension and flexion, and then ankle dorsiflexion. So ankle dorsiflexion is ability to move your foot upwards. So the improvements in myotonia and improvements in strength are really looking at single dimensions of muscle activity. And so they're, I hesitate to say simple, but they're simpler measures of muscle function because we're just looking at one particular aspect. If we look down at the bottom of the slide, we can see now we're looking at mobility measures. And when we test different aspects of mobility, what we're looking at is how improvements in myotonia and improvements in muscle strength translate into more complex actions and in where you need coordinated contraction and relaxation of multiple different muscle groups in order to perform the task. So this really tells us how the more simple measures of muscle function and more simple improvements that we can measure one at a time, how those add together in more complex tasks. So one of those on the left here is the 10-meter walk run test. So essentially we're asking participants from a standing start to walk 10 meters, and we measure how long that takes and what we express the data in terms of change from baseline. So if the baseline took 10 seconds and then after they receive drug it takes 7 seconds, then the change is a negative 3 seconds. And then on the right-hand side, we have what we call the timed up and go, or the tug. So the participant starts in the seated position. They're asked to stand up. They walk 3 meters. They turn around. They walk 3 meters back and sit down again. And this is really quite a complex action, and it's something that we all have to do every day, many times. I think I've lost count of the number of times I've got up from my seat, walked somewhere, walked back and sat down again today. So it's definitely something that I have to do dozens of times during the day, and these kind of activities become progressively difficult as DM1 progresses onwards over time. So if we can move to the next slide. Okay, so I'm now going to hand over to Dr. Johnson to walk you through the actual clinical data, which is the really exciting part of today. And so over to you.
Great. Thanks, Steve, and thanks everybody for tuning in today. Really nice background and overview, so I think we can go ahead and move on to the next slide. So this is probably the busiest slide I have, but this is a summary of the characteristics of the individuals that participated in the trial. And so I'll draw your attention to a few things. Number one, you can see that both the placebo and the 4 milligram, or the highest dose cohorts, were a little bit older and generally a little bit more severely affected. Predominantly women enrolling in this study. The spliceopathy score is a way to aggregate and measure the amount of RNA missplicing that occurs because of that repeat expansion. And so you can think about this as a composite score of the effect of the repeat expansion in the cells, with 100 being the worst and 0 being the best. And so you can see there, again, the spliceopathy score a little bit worse in that 4 milligram and placebo. And then likewise, a little bit more myotonia in that high dose cohort and a little bit slower getting around on the 10 meter and timed up and go. We can go to the next slide. So this is a summary of the adverse events. We talked about this a little bit throughout. So you can see the most common adverse events are expected. So procedural pain, because we do biopsies and other procedures. And then anemia, just because of the way that this oligo gets into the cells, as was mentioned earlier. There are two serious adverse events. One that was mentioned at the outset, resulting in that partial clinical hold. So this is the technically called the bilateral ischemia in the region of the lateral nuclei and the thalamus with some sort of hemorrhage, but you can also think about this as a stroke in the area where the thalamus was, with some bleeding shortly thereafter. And this is a serious disease, so there's one serious event that was unrelated to the treatment, reaction to opioid pain medication following a surgery. Next slide. This is a summary of the biomarker data here. So you can see that the grouped against the placebo is the very far left column in each graph. And then you can see the amount of DMPK reduction, which is what we want to have happen in the 1, 2, and then 4 milligram cohorts in different shades. And then the parallel to that is that 22 gene composite source. So again, this is the score measuring how much missplicing or how much that repeat is affecting your proteins to make the right shape, or your body to make your cells make proteins the right shape. And so you can see on average it's about a 42% reduction in DMPK RNA. And then you can see that both in the 2 and 4 milligram dose cohorts, there were a number of people who had an improvement in their RNA splicing. So 0 being no change whatsoever. And then you can see that, minus 10 change, minus 20. And I said, basically this scale is on a 0 to 100 score. So that kind of gives you a feel for the magnitude of the change. Next slide. Okay, so then let's talk about the myotonia. If you open your hand and squeeze it and then try and release it, you can see that the myotonia, that's part of the myotonic dystrophy, makes it difficult to open your hand. That myotonia is also some of the reason why people can have trouble with stomach upset or difficulty swallowing or a number of other symptoms that are related to the disease. So let's go ahead and play this first video. So this is somebody being asked to squeeze their hand and then open it as quickly as they can. And you can see there in synchrony, the baseline versus that 3-month dose and 3 months after that third dose. And you can see there's a clear and visible improvement, 3 milligrams. This is at the 2 milligram dose. Next slide. Okay, so that's the video. But if you took everybody and took their videos and timed it, how long it took to open the hand and release it, this is what those data look like. So these are, you can see here the video hand opening time. You can see the placebo at the top didn't change very much. You can see the 2 milligram cohort does improve a little bit, but then returns back by the end of the study. And then you can see for that 4 milligram cohort clearly separating in terms of the change in myotonia from baseline across all the participants in that group. And that is statistically significant. So that's a clear sign that the myotonia is being improved, particularly in that highest dose cohort. Next slide. All right, so that's the myotonia. Let's talk about strength. So here, if you've been to your neurologist and you've watched them push and pull on your muscles, and they can say, well, you're weak in your ankles, they're weak in your grip, you're weak in your hips. This is a more quantitative way of measuring that same idea. So they quantitate the amount of strength you have in your hand, your elbow flexion, elbow extension, knee flexion, knee extension, and ankle dorsiflexion. So all muscles that can really be affected in myotonic dystrophy. And then they're normalized against a control group for a percent predicted. So for you, for your age, for your weight, for your height, et cetera, what is your percent predicted in those strengths? So if you take that composite measure, you can see what it looks like. And you can see a separation, particularly at that day 183 from the 4 milligram to the placebo. and that 2 milligram dose cohort splits the difference. So I think for somebody that looks at these data, it's nice to see that there's a clear difference in terms of the doses, in terms of the amount of effect that you can see, but definitely an improvement in strength, a change from baseline in terms of the percent predicted. And then if you look at the 10-meter walk run, so again, walking 30 feet, here you can see that the 4 milligram cohort separated out from the placebo and the 2 milligram dose cohort. So a suggestion that it's going to improve mobility. And last but not least, next slide. Here looking at that timed up and go, so how quickly you can get up and go, as Steve discussed, and you can see again the 4 milligram dose cohort with an improvement from baseline compared to the placebo and that 2 milligram dose cohort. So early suggestions of improvement in mobility measures, which is so important in this condition. Next slide. And then last but not least, this is the same basic idea. So you can see somebody being asked to squeeze their hand, and you're going to look at this as the 4 milligram dose cohort, and this is the baseline and then 3 months after the third dose, and we can play that video. So they're saying squeeze and then release.
And open.
So you saw the summary data, but I think it's just as impressive and helpful to see, you know, in real life what it actually looks like during this course of the study. So clearly an improvement in myotonia. All right, next slide. Just to wrap up, this is an under-recognized, progressive, and often fatal disease with a high unmet need. I'm excited to be here with the Avidity team, demonstrating that this can be delivered to muscles, that it results in splicing improvements and DMPK reduction, and that that then translates into things that we care about. So directional improvement in multiple clinical endpoints at the dose range of 2 to 4 milligrams, including improvement in myotonia as early as 6 weeks and sustained, improvement in strength observed at 6 months, and then early signs of mobility and improvement at 10 meter and tug, generally favorable safety and tolerability. And so I think all told, excited to be seeing this move forward into a phase 3 clinical trial. And with that, I will turn it back to Sarah.
All right, I'm coming. I'm just figuring out my camera. There we go. Awesome. Sorry about that. It's like when you're clicking on a button and I think I was muting and unmuting it again in rapid succession. So just before we get into the Q&A, I hope you've got a sense around the work that and share our excitement for the data that we've seen from MARINA, as well as the work that we've been doing here at Avidity. If we go to the next slide, please. So I'm going to close on next steps, and then pretty shortly we'll get into a live Q&A. Firstly, you know, for MARINA and for AOC 1001, the company was founded on the aspect of bringing meaningful therapies to people who are living with devastating rare diseases. We are steadfast in that commitment, and to do so and to advance our programs. I also want to add again that we are acutely aware that we are part of the community here, and that this takes a large village, both from our team here at Avidity, the study participants, their families, our advocacy partners. We are so grateful for the work of the Myotonic Dystrophy Foundation and others in setting up the NDM1 study and the DCRN network, and with our partnership and the work with study investigators and their teams who have worked so hard and are so committed to get us to where we are today. The open label extension study is ongoing for people who were previously enrolled in MARINA, and we will continue to do that. And we'll actually have our first look at the data from the MARINA open label extension study very much towards the end of this year. For us as a team here at Avidity, the data that we've seen from MARINA, even more so reinforces and increases our enthusiasm on the potential for AOC 1001 to ultimately be an effective treatment for people living with the devastating impact of myotonic dystrophy. As one of the aspects that we'll be doing next with the data package is getting into discussions with regulatory agencies around the design of a pivotal study. So a pivotal study, just from a terminology perspective, also called a phase 3 study. It's really called a pivotal because it's the study that then you then move to seek registration approval for. And we're partnering with the advocacy community to get you more information and resources as soon as they become available. And I'll close with just a little picture of who we are as a team. So this is some of the team. It's not all of the team, but just so you know, these are real people. These are real humans who are awesome and incredibly committed. And at its simplest form, we're people looking to bring great science and ultimately therapies to other people. And we're looking to be able to make a difference in people's lives. That's what brings the team here at Avidity to work every single day. And I'm so grateful for the team here. I think in thanking everybody else and all the community, I forgot the team here at Avidity. So we're so grateful for the team here. And with that, let's get into Q&A. And Alyssa, I believe I am handing over to you.
Great, thank you so much, Sarah. And I'll also invite Steve and Dr. Johnson back to come on camera to answer questions as well. Just a reminder that you can ask questions live. All you have to do this is open the questions tab and type your question and click send. Also, if you're using a smartphone, you can click on the question mark icon at the top of the screen and type your question and click send. And we have already received a number of questions both live today and during your registration. So we thank you so much for your engagement, and we really look forward to answering those for you that we haven't already addressed. The first question is actually, we received several questions around the next steps here. So I'll ask Steve to answer this. I know we spoke a little bit to it during the presentation, but Steve, if you can really talk about the plans for future clinical trials, really what comes next after this top line data is being shared?
Hopefully I'm unmuted now. So the next step for us is really going to be what Sarah referred to as a pivotal study, so phase 3 clinical trial, and we're working through what that could look like now based upon the data. The data that we've seen so far, as I mentioned earlier, is top line data. We don't have all of the data from the study yet, so we're as the additional data comes in, we're looking through that, and also looking at the data from the NDM1 open label from the NDM1 natural history study to help inform those things as well. There is a step that occurs ahead of getting into a phase 3 trial, which is to find a path forward with FDA, both in terms of lifting of the partial clinical hold and agreeing around a phase 3 study design. And these are all things that we're continuing to work on right now.
Great, thank you, Steve. We also received a lot of questions around the meaningfulness of the efficacy results that we looked at from the top line data. So Dr. Johnson, if you could help us contextualize what the data means in terms of muscle strength and myotonia.
Happy to. So both from clinical experience and also from surveys that have been done, like the CRISPR project or out of the University of Rochester, we know from large groups of individuals living with myotonic dystrophy that difficulties with hand function, difficulties walking, getting up and going around, and then mobility or falls are all big deal problems in this condition and cause lots of problems being able to keep your job, being able to do things that you want to do with your life. So what are some of the ways that we can take that information, you're saying these are things that are important, And how can we measure that in a clinical trial and show that we can measure it over a short period of time? Well, so the myotonia really captures difficulty using your hands, opening beers or bottles. The timed up and go and 10-meter walk run also capture things that might be associated with like falls or difficulty with walking. And then the strength just is kind of a core part of that. The lack of strength is what drives a lot of those other problems. So I think all told, the things that we showed today really hit on things that if you took that back and tried to, open your car door and get around, you would find this to be a big help.
Great. And the second part of the question around efficacy is around the effect on different symptoms or manifestations that might be addressable by AOC 1001. So do you see any effect, or can we expect to see any effect on cardiac manifestations, respiratory manifestations, and even swallowing?
Yeah, that's a good question. As Steve mentioned earlier, we know that this drug doesn't get into the brain. So that's one thing that I think we're not particularly looking at, but I will say, as was mentioned multiple times, there's a whole bunch of information that was collected in this trial about heart function, respiratory function, gastrointestinal function. And so those data are just going to, I think that they're going to come later. I would say because we saw changes on the splicing index and because we saw changes in the underlying problems, so the reduction of DMPK, and we know the drug can get into those other organ systems, I would hope that we would continue to see benefit there, but some of those other organ systems are a lot harder to measure than, say, for example, your myotonia or strength.
And the next question is for Sarah. In thinking about continuing development of AOC 1001, there's questions around availability for testing for children or the pediatric population with myotonic dystrophy type 1, as well as if there's any research being done for myotonic dystrophy type 2.
Yeah, so I think, first and foremost, obviously, we're incredibly excited around the data that we saw in MARINA. We are very committed around being a long-term partner in the for the myotonic dystrophy community. So one of the areas is, other areas that we would be looking at over time would be pediatric and some potential other diseases as well. But it's still early from that aspect. So it's difficult for me to really say when that would be. We're also very focused on the immediate next steps, which is around getting the rest of the data from MARINA and then working through with regulators. But it is very much top of mind.
Great. There's also been some questions around the administration of AOC 1001. And so, Steve, if you could answer this, how is AOC 1001 being administered and how often will this drug need to be taken?
The drug is administered as what's called an intravenous infusion. So it's administered in a clinic setting, and the participants need to have a cannula put into one of their veins, and then the drug is given as what's called an IV over a period of a couple of hours. So that's how it's administered. And then the frequency as we move forward will be determined by the dose. So as we saw in the data that Dr. Johnson presented, at the 4 milligram per kilogram dose level, we see that the treatment, the improvements that are seen are maintained over about a 12-week interval. So from day 92 through to day 183, we didn't see really attenuation of the treatment effect. Whereas at the 2 milligram per kilogram dose level, we saw that the treatment effect was wearing off towards the end of that 12-week interval. So for 2 milligrams per kilogram, we know now that we need to dose that more frequently than every 12 weeks. And we're just working at the moment within our in-house group to model what would be the most appropriate dose interval for that dose.
We've also received a number of questions about getting involved in clinical trials. So Dr. Johnson, can you speak a little bit about how folks from the community can really think about signing up for clinical trials, and if there's anything they can do now as they prepare for that?
Yeah, I mean, I think this is just such an exciting time for the entire community, because I think there's going to be a lot of choices out there for people who want to get involved. So you can go to the Myotonic Dystrophy website, which you did because you registered for this webinar, and look there at their clinical trials toolbox to look for studies. We have Myotonic Dystrophy Clinical Research Network sites. There's now 22 across the world. Many of those sites will have a good handle on where to access clinical trials, if not at that site specifically. And then you're always welcome to reach out, I think, to the foundation by email or on clinicaltrials.gov. So lots of choices.
Great. And I know we've gone over time, so maybe one last question. And if there are any questions following this, you can always reach out to Avidity at patients@aviditybio.com, and we'd be happy to continue to answer your questions. So in the interest of time, our last question I'll direct to Sarah. What do you need to advance your research in myotonic dystrophy?
I think, probably the biggest element around that, and I spoke to this at the beginning, as with any important things in life, it takes a village. And that we will not be able to do this on our own, and that a partnership with investigators like Dr. Johnson and the team around those sites, partnership with the foundation, who have been so instrumental in getting us to where we are today, and then also partnership with the patient community. They're the key things that will help us move forward. The team at Avidity were incredibly committed. We're very excited around what we've seen and around being able to ultimately bring a drug forward to people living with this disease. But I think the key to anything is that partnership and that collaboration that we hold together as a community. The most difficult things in life are never done on your own. They're always done as a team, and they're always done as part of a community.
Thank you, Sarah. And with that, I'll turn it back over to Tanya to close out our webinar.
Thank you so much. Thank you to Sarah, Steve, Nick, and Alyssa for being here today. This is incredibly exciting data. We appreciate the details of the functional assessments and where improvement was identified, really, as well as how the medicine is administered. We're grateful for the consistent communication with our community. Really appreciate your partnership. We look forward to the next phase of the trial and want to thank everybody for their time, their energy. We are going to post the video to our digital academy as soon as it's available. And thank you, everyone. Have a fantastic week. Take good care.